Okay, so next up is Daša Lipovšek, and she is the vice president and head of lead discovery at Aktis Oncology, where she's responsible for the discovery of target binding mini-protein leads. And I think she's been developing these into radiopharmaceuticals. Throughout her years in biopharma, she has combined structure-based design and in vitro evolution to target binding proteins for use as therapeutic and diagnostic agents. Her particular emphasis is on design single-domain proteins. So I will also add, because the three previous speakers are all faculty at MIT, we are also claiming Daša. She got her BS in biology from MIT and a PhD in chemistry from the University of Cambridge. So she's going to tell us about mini-proteins for targeted radiotherapy. Really looking forward to it.
Thank you. Yeah, so many thanks to the organizers for inviting me, especially since I will not be talking about antibody-drug conjugates. I'm somewhat reassured by the talk so far after lunch, as they got more and more exotic. So maybe it's not going to be such a huge jump to leave antibodies behind for a moment. So what are mini-protein radioconjugates? At the functional conceptual level, they're very much like antibody-drug conjugates. They are made of a binding piece that has a high affinity and high specificity for tumor-associated antigens, and that delivers the molecule to the tumor. The binding piece, the binding domain, is covalently linked to a linker, which in our case has a metal chelator, and the metal chelator coordinates the payload, the cytotoxic payload, which in our case is a nucleotide. A radioisotope, radionuclide. And this can be a lethal beta or alpha emitting radioisotope, such as actinium, which is what we like at Aktis, or it could be a gentler imaging diagnostic nuclide, such as copper or gallium. At this point, at this high level, the similarities end. The size of a mini protein is probably less than 5% of an entire IgG. There's no such thing as CDRs. Pretty much the entire mini protein is responsible for binding, because part of it will be in direct contact with the target, but the rest of it is so close that a small change across the structure is going to change binding properties. So to compare the mini proteins to the rest of biologics, put them further in perspective, a quick chart here that I'm not going to examine in great detail just puts them on the continuum between linear peptides and IgGs. And you can see that they are much smaller than IgGs, but they are larger than linear peptides, macrocycles, or bicycles. And they are large enough to have a defined secondary structure and a folded tertiary structure. So you can indeed generate three-dimensional stable surfaces to interact with such surfaces on target proteins. So you do not need a professional peptide binding target in order to bind a mini protein. We were delighted to find, after we started the company, that we can indeed reproducibly select very high affinity mini proteins. Our first two, our clinical compound and our compound that's heading into the clinic, above 200 picomolar in affinity. They're also very specific. What's very different about proteins in this size range versus IgGs is that their half-life in the bloodstream is much shorter. They get filtered out through the kidney, so the exposure to most normal tissues is much lower than for IgGs. And when you're carrying something like an alpha emitter as your cytotoxic part, it's really important to clear out of normal tissues as fast as possible, and especially not to expose bone marrow. Of course, I have mentioned renal filtration. This means that we have to be particularly attentive to possible toxicity to the kidney. So yes, you can filter through the kidney, but the molecule cannot be picked up, internalized by kidney cells, because if it gets stuck there, you can irradiate the kidney and cause real damage there. This is a summary of our most advanced molecule, AKY-1189, which was selected to bind to nectin-4 a target associated with urothelial and several other solid tumors, including triple-negative breast tumors. So you can see that starting with a hit in 47 micromolar range, we managed to affinity mature using the marvelous technique of yeast surface display, as well as more traditional medicinal chemistry. We matured this molecule down to 220 picomolar. Another difference between mini-proteins, at least our mini-proteins, and antibodies is that we do not use recombinant expression systems to make them. They're all made by peptide synthesis, so purely chemically, which means that in advanced stages of optimization, we can use non-canonical amino acids and further tweak their properties. The middle panel here shows us that going from the second to last molecule we described here to our clinical development candidate, we improved the binding and distribution and localization to the tumor, and at the same time reduced the amount of uptake by the kidney, which very much improved the predicted safety of this molecule. Looking at mice with xenografts that express Nectin-4, you can see very effective suppression and shrinkage of tumors, and that is dependent on the amount of Nectin-4 expressed on those xenografted tumors. So a normal type of expression that matches most patients gives us very efficient, complete suppression and melting away of the tumors, but a very low expressing xenograft is not as well inhibited. So again, just another way of demonstrating that the activity of these mini-proteins is indeed dependent on the binding to the target. Of course, nice as it is to be able to cure mice of tumors that we gave them on purpose, it is really the bottom line is, can these mini-proteins end up in the right tumors in patients, and can they help cure people of cancers that are dangerous to them? So I can share with you the first inpatient imaging studies. On the left is a patient with urothelial carcinoma, and then on the right there are two breast cancers. And all of these images were taken either with 1 hour after injection, so before all the normal tissue distribution got cleared. But what we can see is that in these bright spots, which are metastasis of the tumors, is that the mini-proteins can penetrate a large number of different tissues and end up in the tumors. After 3 hours, we even see them in metastasis in the brain, and that's probably because the brain-blood barrier had degraded in this patient due to the advanced nature of the disease. At this moment, well, actually, about a month ago, we announced the start of our first phase 1b clinical trial with this molecule in urophilia and other Nectin-4 positive tumors. So we are waiting with bated breath to the results of those studies. And I'm happy to see that we will have 4 minutes for questions.
Thank you so much. And I really liked your analogy that this session is really, we're going outside our lane, but sometimes you get there faster.
Thank you. This is an exciting talk. Are you concerned about anti-drug antibodies since it's a novel protein?
Not particularly. We're thinking about them. There are several advantages of this platform from the point of view of anti-drug immune response. One is because the molecules are so small. This one is 45 amino acids. Just by chance, there will be fewer T-cell epitopes in them. We do use in silico prediction of T-cell epitopes, and since at every in vitro evolution step, we have multiple molecules to choose from, we choose the ones with the lowest predicted liability, and this one looks very clean. The other reason why we're not that concerned is the doses here are very low, typically about 10 micrograms per patient, because the radionuclide is so lethal, so you can only need 1 molecule per a couple of cells of tumor. And then you can also think of it this way. Let's say that you're an antigen-presenting cell and you grab onto one of these molecules, you'll get fried. So it's a lower risk than you would imagine.
Thank you.
Okay, we'll take a question over here.
Hi, great. You mentioned the importance of reducing kidney retention in your molecules, and you showed the first one had really high, and then you got low. I'm curious, did you have to do iteration, in vivo distribution iteration to get that? How did you approach the reduction in kidney?
The simple answer is yes. We did a lot of engineering, and the 805, the one that I showed you, is nothing. The very first low nanomolar binder just lit up the kidney like a Christmas tree. So there's a combination of in vivo and in vitro work. We have a cell-based assay that mimics to some degree kidney cell uptake. So we also have an AI model that we've trained with many compounds, so that we do have some in vitro and in silico tools, but in the end, you go into mice. So far, we've seen good translation between mice and higher organisms. But it is, this is definitely the one area that you have to really pay attention to.
Okay, we can take one more question.
Very quick. What's the advantage of the mini protein over the in vivo discovered nanobody and humanized nanobody? Thank you.
I think in terms of pharmacokinetics and clearing fast from normal tissues, I think they're quite similar. I think the likely advantage of mini proteins is because they're that much smaller, you would expect them to penetrate tissues faster. And in terms of at the discovery stage, we select the best binders, let's say in yeast display. We have a set of sequences. We feed them into a peptide synthesizer, and a week later, we have our molecules to work with. So you don't have to go through making DNA constructs, starting cell lines. So the iteration time for discovery is faster. And then the third thing is that we can use non-natural amino acids that give you more tools, so larger alphabet. One could argue that if you had an antibody domain, you wouldn't need non-natural amino acids because you have such an effective natural system. So maybe that's a wash. But we like them. And from my point of view as a protein engineer, they're incredibly easy to work with. You can understand how the different parts interact. You can make, for using yeast display, you can make genes in one piece. You can define exactly what every amino acid is going to be that you're going to test. So they're really just a dream to work with. But I think the bottom line, I have to keep reminding myself, this company was not put together to entertain me. It's about curing patients. So we'll have to see what the advantages are once we finish clinical trials.
Thank you.
So let's thank Daša again. And then I really want to thank all the speakers this afternoon for these really great snapshots of their beautiful work.